So why do we care about synergizing immunotherapy with molecularly targeted anti-cancer treatment? Well, anti-cancer drugs often compromise crucial components of the adaptive immune response. And so while they produce a lot of antigenic activity as they induce apoptosis of the tumor cell, that activity cannot be properly harnessed by the immune system because it's impaired at crucial junctures where it can, where an antigen-triggered immune response is expected. So one needs to reverse that, and in order to do that, one needs to make drugs sufficiently selective so that they are not going to impair the adaptive immune response. And that's the thrust of this brief talk. So to illustrate these ideas, I will focus on one example, imatinib, the drug known as Gleevec in the market, used to treat chronic myeloid leukemia. This drug has been created by man to inhibit three major anti-cancer related targets: the BCR-Abelson kinase, the KIT kinase, and the platelet-dependent growth factor receptor kinase. But it also has some undesired cross-reactivities, in particular with the lymphocyte-associated kinase, LCK. And that is the culprit for its undesirable side effects, as it impairs some crucial components of the immune system. So in this slide, we can see LCK as a crucial signal transducer in the pathway that the signaling pathway that is related to antigen-triggered T cell activation. And we can see why it is an undesirable target for a cancer treatment that portends to utilize the immune system as an ally of the drug treatment and not antagonize it. To address this problem from the perspective of rational drug design, we don't use the structure itself as the guiding element, but rather the epistructure, what I like to call the epistructure, that is to say the structural features of the solvent at the protein-water interface. A specific feature to be used is the dihydron, which is the solvent-exposed backbone hydrogen bond. And so that forces the solvating water molecules to adopt very uncomfortable hydrogen bonding coordination conformations that make them very prone to be removed. So solvent-exposed backbone hydrogen bonds, the dihydrons, are actually in effect sticky, and therefore can be used as a selectivity filter to, in this case, filter out LCK from the targets of oncogenic relevance. So here we see an epistructural comparison of LCK and KIT that inspires the remodeling of imatinib into an immunosynergic drug.